Good morning, students. This is for third semester, second year biotechnology students. The title of the paper is Biophysical Techniques, Unit 1, Spectroscopy, Lecture 6. In previous class, we discussed about the uh, deviation of Beer's law. The next, in this session, we discuss about the Molar extension coefficient or attenuation coefficient. After completion of Beer's law, so the molar extension coefficient, what molar extension coefficient is derived from Beer's law. Here, when a monochromatic light beam pass through the homogeneous solution, that means when a monochromatic light, it is a cuvette containing a homogeneous solution. Here the monochromatic light is used for the lambda maximum of light. Here when a monochromatic light beam pass through the homogeneous solution, light absorb in the solution. So here absorbance of the light is derived in lambda centimeters. So when absorbed, the solution is directly proportional to the product of concentration and path length. This is otherwise called as A, means absorbance is equal molar extension coefficient, concentration and length. So A stands for absorbance, E molar extension coefficient, C that is called amount of concentration, and L is the path length. So this is, and C and L, these two are the concentration of the solution, and second one is the length of the wavelength. These are determined by the extension coefficient via absorbance. So extension coefficient or attenuation coefficient explain about the interaction between the graphical representation of the concentration versus on x-axis, y-axis on the absorbance, what we measured the optical density in the form of nanometers. Here we can take a graph is obtained by the lambda maximum. A graph is obtained by the lambda maximum. So these are the what we take the concentration of the solution. Here we can explain about the first one is the concentration on x-axis. It is represented by mu c per ml. In y-axis, what we call absorbance. It is represented in the form of nanometers. So here the standard curve is present, the linearity standard curve. These are the different kinds of sample containing the optical density values. Those we are taken from the plot, that means graphical representation through concentration versus absorbance. Then here we can take particular absorbance of the sample containing the concentration of the solution. So the extension coefficient is measured by a homogeneous solution in a cuvette. It is a cuvette containing the solution. Here we can take a lambda maxima, that means the wavelength. Lambda maxima wavelength. In this wavelength to produce absorbance. In this wavelength to produce absorbance by using Beer-Lambert's law, what we learned from the last classes. So this is measured by A is equal to extension coefficient concentration and length of the light. A is equal to ECL. A is the absorbance. A is the absorbance. E is extension coefficient. E is the extension coefficient. What we learn, the molar extension coefficient or attenuation coefficient. C is the concentration of the amount of the concentration in the particular given unknown solution, amount of the concentration on the solution. And L is the path length. L is the path length. So here we can derive the extension coefficient, E, extension coefficient. Here it is the multiplying. It is the multiplying. Where we multiplying, if to take it is the divided by. Multiplication ni అంటే ఇక్కడ మనం చేంజ్ చేసినప్పుడు ఏమవుతుందంటే E is equal A by CL. A by CL అంటే ఇక్కడ A అంటే ఏం చెప్తున్నామంటే what we call is absorbance. A is the absorbance. C means amount of concentration. L is the path length. L is the path length. Here the path length is derived from here the standard plot or standard graph what we take. So in this standard curve, so x-axis containing the concentration of the compounds what we take either estimation of protein or estimation of DNA, estimation of RNA samples we analyze by using x-axis. On y-axis we will take the absorbance according to the nanometers, like 400 nanometer or 430 nanometer, 520 nanometer or 610 nanometer, what we take the particular wavelengths of the nanometers. These we will take this type of nanometer according to respectively concentration of the sample belongs to the color representation also. So ikkada determining the every chemical and wavelength having particular wavelength, every chemical and particular wavelength of the selected selected wavelengths of on the monochromatic filters. Here 6 to 8 type of different kinds of filters are present. By these filters we can choose a single type of filter is called as monochromatic light. That type of monochromatic light exhibit particular lambda, that means wavelength, focus on the focus on the what we call the cuvette. In this cuvette containing a homogeneous solution. On that homogeneous solution satisfying the Beer-Lambert's law. Here the biological samples converted through electrical samples by using the layer that is called photocell that containing selenium layer is present. On that selenium layer biological signals converted into electrical signals through amplifier. By that amplifier we can know the optical density by using the galvanometer. These type of values we can produce by using extension coefficient. This extension coefficient is represented by E is equal absorbance by concentration of the solution and path of the length. Here the corresponding plot is called lambda maximum wavelength selector, by what we choose the concentration of the samples. concentration of the samples. Then, so the finally molar extension coefficient can explain the graphical representation of the sample using the calorimeter or spectrophotometry. According to Beer-Lambert's law, here the concentration versus absorbance. Concentration on x-axis, absorbance on y-axis. The concentration versus absorbance through graphical representation to produce a plot. In this the linear plot may not produce by using that sufficient, by using the values. Here the deviation may occur. In this deviation is called Beer's deviation, what we learned from last class. Here some values may decrease, some values may increase according to their color intensity. So here finally we conclude the extension or attenuation coefficient. These attenuation coefficient may produce three categories. The first one is the absorption coefficient or attenuation coefficient, and the second one is the mass attenuation coefficient. Third one is the molar extension coefficient. This molar extension coefficient measurement how strongly a chemical attenuated light of the specific wavelength. So next, it is an intrinsic property of the natural intensity property of the material, that is nothing but a chemicals. The intrinsic property of the material. The SI units or molar extension coefficient is square meters per mole. It is the unit of the molar extension coefficient. In biochemical calculation, the molar attenuation coefficient of a protein exclusively depend upon the number of aromatic amino acids, particularly upon tryptophan. Aromatic amino acid. According to the importance of the molar extension. Importance of the molar extension coefficient. So here the importance means measurement of how strongly a chemical species absorb light at a given wavelength. So here measurement of how strongly chemical species light to the absorbance of the given wavelength. Particular wavelength లో ఎంత అబ్జార్బెన్స్ బేస్ చేసుకుని ఆ లాంబ్డా లైట్లో ప్రొడ్యూస్ అవుతుంది అనేటటువంటి దాన్ని ఫస్ట్ మనం దీంట్లో ప్రొడ్యూస్ చేస్తాం. ద సెకండ్ వన్ ఈజ్ ద ఇంట్రెన్సిక్ స్పీసెస్. That means it is an intrinsic property of chemical species. It is depend on chemical composition and chemical structure of the material. So second one is the intensity of the specific chemicals, the chemical structure and properties, and having the ionic groups and the van der Waals effects. So likewise the chemical properties and the interaction between two concentration versus absorbance. The third one is measurement of absorbing is very fast method. So here the measurement of absorbance is very fast method for determining the chemical concentration, and species in the solution must be given. And the fourth one is the it is other methods. What are the other methods? If not their absorbance is not determining in the whether spectroscopy, what are the other methods of the measurement of extension coefficient is the other measuring methods are the titration methods. Here the titration methods can be titration methods can more time it requires additional chemicals. It having more time and it requires additional chemicals. And the SI units of the extension coefficient is meter square per centimeters. Here the extension coefficient here the extension coefficient is the meter square per moles. Centimeters kadani mole. It is the extension coefficient is the meter square per mole. Next, estimation of molar concentration solution from its measurement of the absorbance. So this is the short answer question. Extension coefficient or attenuation coefficient, molar extension coefficient. దీనికి సంబంధించిన నోట్స్ మీ యొక్క వాట్సాప్ గ్రూప్స్లో దీన్ని నేను మీకు అప్లోడ్ చేస్తాను. ఇఫ్ ఎనీ క్వరీస్ ఆర్ డౌట్స్ ఏమైనా ఉంటే కనుక కాంటాక్ట్ మై నెంబర్. థ్యాంక్ యూ స్టూడెంట్స్.